Designing a transgenic virus to engineer wild mosquitoes with resistance to Zika virus. Before we dive into our experimental design, we would like to start with background information on the Zika problem. Zika is a mosquito-borne virus carried specifically by Aedes mosquitoes, notorious for carrying dengue and yellow fever. Zika was first found infecting humans in the 1950s in Central Africa. As the virus was linked only to mild symptoms and localized to undeveloped regions, there was little international interest. It wasn't until 2015 that Zika shot to international headlines as the virus unexpectedly spread outside of its endemic regions, becoming a worldwide problem. This was compounded by the fact that Zika was linked to birth defects, including microcephaly. In the past, scientists and public health officials have attempted to fight mosquito-borne disease via a multitude of approaches, all of which have limitations. First, male mosquitoes have been created which only have male offspring. This results in a massive decline in mosquito population over several generations. This approach effectively reduces the number of mosquitoes but has upstream ecological consequences for animals that rely on consuming mosquitoes. Second, mosquitoes carrying Wolbachia pipientis bacteria have been released in major cities and infected areas. Wolbachia is highly infectious to mosquitoes and is able to reduce host fertility and inhibit the replication of the Zika virus. Though very promising, the long-lasting effects are unknown. Furthermore, wild mosquito populations appear to acquire resistance to the bacteria over time. Third, vaccine development is in progress and is an effective tool. Protection relies on vaccination, which requires organization and continued government investment. This can be difficult for many poorer countries. Another problem is the fact that a vaccine will not eliminate a virus like Zika, which has wild reservoirs. Our approach takes these limitations into consideration. By editing the genomes of wild mosquitoes using a transgenic virus, we hope to have a limited ecological effect and provide long-lasting Zika deterrence. Our approach to this problem began with choosing a target specific to the Zika virus. After finding this target, we designed a system to attack it. We searched for an integrated mosquito virus to act as a vector for adding the system to the mosquito genome. The integrated virus was genetically engineered to include the genes for the Zika attack system. The transgenic viral vector was mass-produced. Finally, the viral vector was packaged and released into the wild environment. Step 1. Finding a Zika-specific target. The Zika virus is a positive-sense, single-stranded RNA virus, which can be directly translated in the host cell. Literature searches show that a complex 3D structure forms within the viral genome when it is in the host cell. This complex 3D structure is known as subgenomic flavivirus RNA, or in short, sfRNA, and is composed of many stem loops. This viral sfRNA protects Zika from host defenses and allows translation of the viral genome. Cleaving sfRNA would allow the host to degrade the Zika virus genome. Hence, sfRNA is an excellent specific target. Inside of mosquito cells, the Zika virus is attacked by XRN1 exonuclease, a defense mechanism of the host against foreign DNA. XNR1 cleaves along the Zika viral genome and stalls upon interaction with the stem loops of sfRNA. This stalling allows the viral genome to unwind and be translated within the mosquito cells. This effectively activates the viral pathogenicity. If the mosquitoes are modified so they can cleave the sfRNA, the XRN1 exonuclease will be able to degrade the entire virus. This eliminates the virus and hence prevents pathogenicity. Step 2. Finding a system that can attack sfRNA. CRISPR is used by bacteria as a defense against exogenous nucleic acid. This makes CRISPR perfect for targeting viral genomes. Class II type II CRISPR, isolated from Streptococcus pyogenes, is the system typically used in bioengineering. The Class II system is composed of several CRISPR-associated Cas proteins, along with coding regions for scaffolding tracer RNA and guide CrRNA. The Cas proteins include an endonuclease Cas9, which cleaves dsDNA. Tracer RNA binds to Cas9 and CrRNA, which localizes the endonuclease to a complementary region on the exogenous DNA. As Cas9 is only able to cleave dsDNA, Class II type II CRISPR won't work for targeting Zika, which is a ssRNA virus. To accommodate the type of virus we're targeting, Class 2 Type 6 CRISPR isolated from Leptotrichia sahi differs from Class 2 Type 2 in two ways. First, Class 2 Type 6 CRISPR doesn't require a scaffolding tracer RNA. Second, Class 2 Type 6 CRISPR has a C2C2 ribonuclease which cleaves ssRNA in place of the dsDNA, cleaving Cas9 protein. C2C2 can bind directly to CrRNA, which directs the ribonuclease to the site on the RNA which is complementary to the CrRNA. Cleavage of RNA will then occur at this site. The fact that the CRISPR Class 2 Type 6 system directly targets ssRNA makes it ideal for targeting Zika sfRNA. sfRNA is difficult to target as much of the sfRNA region is bound in complex 3D dsRNA structure. To deal with this, we analyzed the sfRNA region of the Zika RNA in 3D space using CLC Workbench. CLC Workbench is a bioinformatics tool allowing visualization and analysis of DNA, RNA, and the protein. This program allowed us to visualize the complex structure of the sfRNA. An exposed GC-rich stem loop was found between residues 340 to 440 of the sfRNA, allowing for the possibility of attack with the C2C2 CRISPR system. The sequence of the vulnerable region of sfRNA identified by CLC Workbench was entered into the CRISPR Genome Engineering Resources website to find potential CrRNAs. The output from the CRISPR Genome Engineering Resources website lists all possible 23 base pair guide CrRNA sequences. Each sequence is given a score based on uniqueness and binding strength. The guide sequence with the highest score was selected. The guide sequence will localize the ribonuclease C2C2 to the vulnerable site of the sfRNA, allowing cleavage, resulting in degradation of the Zika virus. Step 3. Finding an integrated mosquito virus to use as a transmission vector. We wanted to use a virus that integrates its genetic information into the Aedes mosquito genome as a transmission vector. This would allow the CRISPR system to be passed to wild mosquitoes. In order to find this, we used high-seq Illumina sequencing to sequence 100,000 Aedes aegypti mosquitoes. All mosquito genomes were then aligned in order to find a common integrated virus. When A. aegypti genomes are compared, most of the sequence aligns perfectly. Regions of difference unique to only some of the mosquitoes indicate a possible integrated virus. We chose Illumina sequencing because of the large number of genomes we had to sequence. Illumina is known for offering high throughput, owing to its massively parallel sequencing capabilities. Mosquito genomes were aligned using muscle multiple sequence alignment, a bioinformatics program for sequence alignment. Sequences that were not common to all A. aegypti genomes were considered as potential integrated viruses. These sequences were searched in the NCBI viral database. After sorting through all the data, a single virus, TTFCU, was picked, as it had properties favorable to our experiment. TTFCU is a linear dsDNA virus. TTFCU integrates into the DNA in the ovaries of A. aegypti mosquitoes. As a result, it is passed on to the offspring. TTFCU integrated DNA is present in all cells of the offspring of an infected host, making it a good candidate for delivery of transgenic genes like our CRISPR system. TTFCU remains integrated in the mosquito indefinitely, being passed from generation to generation. It was found that exposure to DDT can cause TTFCU to excise from the host and package into virion particles. The packaged TTFCU leave the dying mosquito and remain stable until they can infect the ovaries of another A. aegypti mosquito. To genetically alter TTFCU, we must first isolate it from host cells. Mosquito cells assumed to be infected with TTFCU are treated with DDT, which induces viral packaging. Insect cells are then lysed and purified via centrifugation to remove all the large contaminants, like chromosomal DNA and cell membrane. Flow-through is then treated with a nonspecific protease which will digest the capsid proteins and other contaminating proteins. To purify the viral DNA, we perform an organic extraction and precipitate the TTFCU DNA. A small amount of purified TTFCU DNA will be sequenced via Sanger sequencing to confirm the presence of the viral DNA. Step 4. Engineering the C2C2 system into the TTFCU vector. We need to insert our Zika attacking system into our viral vector, TTFCU, which will ultimately integrate the system into mosquitoes. From now on, the native integrated virus will be referred to as TTFCU, while our transgenic virus will be referred to as TTFCU 2.0. TTFCU 2.0 will contain the C2C2 CRISPR system, which will cleave the sfRNA of Zika virus and will be heritable when integrated into the mosquito host genome. We entered the nucleotide sequence for wild-type TTFCU virus into the NEB cutter tool to find single cutting restriction enzyme sites. By choosing two sites that are close to one another and enclose an unimportant section of DNA, we can allow directional cloning into the viral genome. Two unique sites were found, corresponding to restriction enzymes BG3 and BSEY1. Both will leave 5' 4 base pair overhangs. Using the two enzymes together, the viral genome is broken into 3 pieces, of which the small, unimportant middle piece is discarded. Knowledge of these cut sites allow us to order synthetic DNA with corresponding complementary overhangs. A 500 base pair promoter from the vitellogenin receptor gene for A. aegypti was found using a literature search. This paper has found that the vitellogenin promoter acts as a constitutively active promoter for expression in transgenic A. aegypti that is specific for ovarian and germ cells. In addition, this paper found that the transgenes are passed to offspring. The 500 base pair promoter for the vitellogenin receptor from A. aegypti was ordered with additional sequences. A recognition site for BSA1, a type 2S restriction enzyme, was added to the 5' end of the promoter. The site was designed to leave a 5' 4 base overhang complementary to the BSEY1 overhang once digested with BSA1. This will allow the promoter fragment to be ligated into the TTFCU virus after its own restriction digestion. A second DNA sequence was ordered containing genes for essential Cas proteins for C2C2 CRISPR, Cas1, 2, and C2C2, along with our previously designed crRNA sequence complementary to the vulnerable region of sfRNA on Zika. The 5' end of this sequence was designed to contain a complementary region to the previously ordered promoter sequence. This region will allow Gibson's assembly. Finally, the 3' end was designed with a BSA1 recognition site, which will leave a 5' 4 base pair overhang complementary to the BG3 overhang. This will allow ligation to the TTFCU virus. The ordered DNA sequence will also contain a gene for GFP surrounded by FRT sites, 5' of Cas proteins. GFP will allow the selection of transgenic cells. It was placed upstream of the C2C2 system to increase the rate of GFP expression. The FRT sites allow for excision of the GFP protein, which is good as green fluorescing mosquitoes don't live long in the wild. The purchase promoter and purchase C2C2 sequences contain a complementary region. When added together with Gibson's assembly master mix, they will assemble into a single piece of DNA. Gibson's assembly can occur at 50 degrees Celsius on a tabletop. The resulting DNA insert contains the ovarian promoter, our programmed CRISPR system with the GFP selectable marker to screen for successful inserts. Recall that NEB cutter was used to identify restriction sites in TTFCU. TTFCU was digested with two unique restriction enzymes, which cut the TTFCU's genome to allow for directional cloning of the insert. The unnecessary portion of the TTFCU genome was cleaved by a third restriction enzyme to ensure that our insert annealed and that the genome didn't self-repair. The sequence from the Gibson's assembly containing promoter, GFP, and CRISPR genes is then digested with BSA1, which will remove the ends of the assembled DNA, leaving sites complementary to the two large fragments from the TTFCU viral digestion. We then mix the two digestion mixtures together and get the viral genome with the engineered insert between the two restriction enzyme cut sites. We add DNA ligase and the gaps will be sealed, resulting in a seamless DNA assembly. This is just a summary slide explaining what the inserted sequence looks like. The promoter is 0.5 kilobase pairs long and allows ovarian and germ cell specific gene expression. It is constitutively active and has an overlap region with C2C2 for Gibson's and has a BSA1 restriction enzyme site. Likewise, the C2C2 insert also has a BSA1 restriction enzyme site and overlap with the promoter for Gibson's. It also has GFP surrounded by FRT sites on both sides. It is important to remember that we added GFP to our C2C2 system so that we can visualize which TTFCU successfully contain our Zika immunity insert. Once selected, the GFP will be removed. Step 5. Mass production of TTFCU 2.0. The next step was finding a way to package the virus in virion particles before mass production. Because the virus was engineered in vitro rather than in vivo, it does not have the cellular machinery to package itself. Therefore, we used insect cells to facilitate viral packaging of our TTFCU 2.0. The process is a little complicated, but essentially we infected insect cells with the wild-type TTFCU and stimulated virion assembly. We then lysed the cells while introducing our engineered TTFCU 2.0, which allowed our engineered vector to essentially steal the capsids from the wild virus. Now that we have packaged TTFCU 2.0 virus, we needed to mass produce this vector before we can release them into the environment. We picked a cell line that would mimic the targeted ovarian cells of the Aedes mosquitoes. This slide shows the final result of our packaged viruses being mass-produced in our insect cell line. The glowing colonies were selected because we know that they contain our TTFCU 2.0 vector that has successfully integrated our CRISPR resistance insert. 81 cells were infected with packaged TTFCU 2.0 cells. 81 cells containing TTFCU 2.0 cells were selected thanks to the expression of GFP protein. Green cells were selected for and cultured. Now that we have selected for successful colonies containing our TTFCU 2.0 vector, we need to remove the GFP indicator. We don't want to risk glow-in-the-dark mosquitoes because they wouldn't survive long in the wild. The GFP indicator was excised from the TTFCU 2.0 vector using FRT-FLP recombinase, a plasmid containing the FLP genes behind a TEDO promoter with constitutively active reverse TEDR transactivator was transformed into our insect cells that contained our integrated TTFCU 2.0. By adding a bit of tetracycline, the FLP recombinase is transcribed from the plasmid and interacts with the FRT sites surrounding the GFP. GFP is excised from our virus. Now that we have removed GFP from our TTFCU 2.0 vector, we can use colony selection again to retrieve our virus, but this time we select for non-fluorescing GFP because those are the ones that underwent successful FRT-FLP recombinase. Our final product of TTFCU 2.0 infected insect cells are mass-produced in a bioreactor. We considered two different types of bioreactor approaches, stirred tank or airlift, and tested these on a laboratory scale first. We concluded that stirred tank bioreactors gave us the best viral growth rate, so we used this approach. For a stirred tank approach, nutrients are constantly pumped in, and a rotor physically mixes these into the AD1 suspension. To balance the input of nutrients, our cells and metabolic waste are constantly removed and the cells are isolated. And of course, we are using low shear axial flow impellers, as well as shear protectorants such as Pluronic F68, to minimize shear force in the tanks to ensure our infected AD1 cells grow optimally. Step 6. Packaging TTFCU 2.0 and releasing the transgenic virus into the environment. After the bioreactor is full of infected insect, DDT is added. TTFCU 2.0 excises from the dying AD1 cells, packaging into virion particles. TTFCU 2.0 is now a fully functioning viral vector, which can be dispersed into standing water in areas known to be high risk for Zika. Mosquitoes that breed in these waters will be infected by our viral vector, which contains the gene for Zika immunity. Once infected, it will integrate into the mosquito genome, including the ovary cells, passing this immunity to offspring. When TTFCU 2.0 virus is released into the wild, it equips wild mosquitoes with the Class II Type 6 CRISPR system, which specifically targets sfRNA regions of Zika virus. When sfRNA is cleaved from Zika, the mosquito will be able to degrade the ssRNA virus, and hence the mosquitoes will no longer be able to spread the virus. As the integrated virus is heritable, it will spread throughout the wild mosquito population, providing long-lasting Zika protection. Finally, we will recap our experimental procedure. First, we began by searching for a Zika-specific target, sfRNA. Second, we found a system to target the sfRNA. Third, we searched for an integrated virus to transmit the Zika attack system into wild mosquitoes. Fourth, we inserted the Zika attack system into the integrated virus. Fifth, we mass-produced the virus. And sixth, we released it into the wild. Finally, here are our works cited. Thanks for listening.